aren't here. Recording in progress. Okay, so this is I think the third class where I've talked a lot about ICZN. And so this is the last sort of major part of ICZN. It's probably the most taxonomic because we're going to talk about types today. It's actually very simple, but just like everything else, our understanding and our use of types has changed. Nice t-shirt over time. Cool, cool, cool. So go back to here. No, come on. There we go. Just to remind you guys. Wow, that's why we're down here. And so types, what a type does is we talk about ICZ covering family, genus, and species. A type is the name of the species in the most simple definition. So this is what we're really working on today is species and species alone. And so when you describe a species, we've talked about this in every class, but I'm going to keep reminding you so that you remember that taxonomy is a real science. Species is a hypothesis. You want your information, you want your research to be replicable. You want to be able to repeat it if you need to. This all comes back to the type specimen. In the end, a species is one specimen. The specimen is the type specimen. And so, in other words, n equals 1, but for a species, it makes sense, n equals 1. But it is a living science and things change over time. So last week, we talked about how seniority, we talked about people describing the same species at different points in time, what happens. This week, when we're talking about types, I'm going to introduce you to 4 or 5 main types, and the use of types has changed over time. Half of the types are to deal with how types have changed over time, if that makes sense. Kenta's one more. Kenta's like one more time. I'm going to introduce to you 5 types. Of these 5 types, 2 types are only to deal with how taxonomy has changed. So I'm going to talk about that. So when you have an original description, when you are describing a new species, in your paper, you must designate a type specimen. You must say, if you have, for example, if you have a Kondo isoginchaku, for example, a Kondo's anemone, for example. If you have 20 specimens of this anemone, you have to choose one. Say this is the type. You have to point your finger. So in other words, when you describe a species, you must designate a type. You can don't wait for me to finish this slide. Yeah, Kisha, I know you're very quick. You can designate more than one type, but I'm going to talk about this after. But one is the holotype. The others are backups. I'm going to talk about this after. I'm going to talk about all the different types and how many to designate and everything. And after that, if you have questions, I'll take them. Okay. So the other thing is, for usually designating a type, usually it's only in an original description. Now, in the past, taxonomy was different, and I'm going to talk about that after. So now, for all of you, 2024, if you describe a species in your paper, you need to say, this is the type. And the different types are what we're going to talk about. The other things, I talked about this in other classes as well, come on, is if you have molecular results and you have one species and there's different genotypes or different clades or subclades or groups, choose your types from one clade or group, because otherwise there's a chance that you have types with mixed species and it makes a mess. For example, if you have a Kenta anemone and some are black and some are yellow, and the molecular signature is different, you should choose all of your types from one of the groups only. Because if you choose from 2 groups, in the future, maybe Kenta Junior comes and changes things. Which is which, you don't know, it's confusing. So always choose your types from one group, okay? Also choose your types carefully. What I mean by this is when you make a species, when you erect a species, when you make a new species, you want your specimen to represent the species well. So maybe it's not too big, it's not too small, it's not weird, it's a very average or normal or good, or it's a good representative, Daihyo-teki. Okay. And the other thing is, I don't talk about this here because I'm only talking about types today, but when you choose your museum, we talked about this in other classes, choose a museum that you think is a good museum, where they look after the specimens, but also maybe in 100 years, the specimen will be there and people look after it. And also think about which country. If you describe a species from Australia, it's good to put a type specimen in a museum in Australia, Japan, etc. But some countries like Palau do not have a very large or well-funded museum. And then you need to think, okay, where do I put things, right? Okay, this is all review, right? We've talked about this before. Let's talk about types of types. This is where we get into the details for today. Often you'll see the word type series. A type series is all the specimens used to establish a species. So you may have a paper with 20 nematodes describing one species. If you list all of those specimens in your materials, specimen 1, specimen 2, specimen 3, specimen 4, those are all, they're the type series. So series means repeated, right? All the specimens that you use to establish a species are the type series. But inside all of these types, inside all of these specimens, there are some types that are more, or some specimens, some types, more important than others. That's sort of what I want to explain. In ICZN, there are 4 types of types that the name, a scientific name, can be attached to. Only 4. The holotype, the syntype, the lectotype, and the neotype. And I'm going to talk about all of these one by one. Okay? Holotype, syntype, lectotype, neotype. Of these 4, 2 are what we normally use, and 2 were created to solve problems. 3, 2. 2 were created to solve problems. Okay. So let's talk about the types. First is holotype. This is the one. This is the most important. Just think, you guys know the name or the word holobiont? If you study corals, right? The one, this is it. This is, if you are erecting a new species, now in 2024, the one specimen that you choose to be the name of the species is the holotype. This, for the species name, is the most important specimen. This is what you want images of from a museum. This is what you want information of. This is what you want DNA from. This holotype, this single specimen, is the most important one. Okay? This is, so if you find something new, you describe it, and you choose one specimen, holotype. So you would say holotype, da-da-da-da-da-da-da-da, and then describe the holotype. Okay? So there's another one out there called syntype. A syntype is a solution for a problem. A long time ago, before 1999 or before 1961, some people would make a species and they wouldn't say what the holotype was. They would just say, here's a new species, here's a list of specimens. But they didn't point the finger. They didn't say, this one is the holotype. So you just have a list of specimens. All of those specimens are called syntypes. Okay? Does that make sense? Yeah? So any of 2 or more specimens, if there's only one, automatically it's the holotype, right? Even if you don't say holotype, if you only have one specimen, automatically it's the holotype, right? But if there's 2 or more, and you don't say this is the holotype, they all become syntypes. The name can be attached to one of these syntypes. It's very messy, but for many older taxonomic papers, in the past, people did not designate types in the 1700s. In the 1800s, they often had just syntypes. The holotype really became must only in the last 30, 40 years. So older papers, often it's just a bunch of syntypes, which makes a problem because it's more than one. So to solve this, actually, let's skip order. To solve this, I should go here. Wait a second, where did it go? I'm missing one. Just a second. This one here. Oh, there it is, my computer's. There's one other type related to syntype. It's called a lectotype. If you find an old paper, and in the old paper, there is no holotype, there are just many syntypes. If there's only syntypes, no holotype, and they don't even say syntype, they just say specimens, because all of these type ideas are newer. You need to choose one to be holotype, because a name is attached to one specimen. If you find an old paper and there's like 20 specimens listed and no one's done any work since then, you can go to the museum, you can look at the specimens, you can choose a new holotype because there was never any holotype before, but this is called a lectotype. So a lectotype is not in the original description, after original description, if the original description has no holotype. So it's like not a replacement, not a new, but an old new holotype. You're coming along and you're pointing the finger, this one. Okay, does that make sense? So a lectotype is created to solve the problem of syntypes and holotypes. Then, hang on, there's another type called the neotype. And the neotype, you guys know the name neo, right? Neo means new, right? So a neotype is when all the original types are lost. And this happens. This happens a lot. The official definition is really, really long, blah, 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 blah. But what it basically means is if someone did a paper and they put all the specimens, the holotype or the syntypes or anything in a museum, and the museum burns down, everything's gone. In this case, you should make, in a new paper, you can make a neotype. But it's not only disasters. There was a disaster in Brazil, right? A lot of the museum specimens were lost 2 to 3 years ago, I think. Almost 5 years ago. See, old guy, yeah. The museum burned down. But also, okay, for example, in Europe, World War II, Japan, World War II, many museums were destroyed. Many types were destroyed. But also, Paris Museum, famous, never destroyed, beautiful. Many specimens just disappear, go missing. It's like a black hole. So if you go to the museum, you look, you can't find any types anywhere, you could designate a neotype. To designate a neotype, what you need to do is you need to find a new specimen of what you think the same species is, as close to the type locality, the depth, etc., as much as you can. The problem is many old papers have no information. This species, this species described from Fiji. That's it. There's a species of Zoantherium described from Manila Bay. Manila Bay is very big and it's now a huge port. Can't do anything. Some species described from Okinawa. So that's why when you describe a species, point name, GPS, latitude, longitude, these things help people in the future. Also makes your research more replicable, right? Okay. There's one more type. Hang on. Paratype. This is the other sort of what I call a normal type. In an original description, when I describe a new species, I have one holotype and I choose some paratypes. And those are the other specimens listed. So it's like a syntype, but it's not. Let's write this down. Holotype is one specimen, right? So if I have a paper with many specimens, I would have a holotype plus paratypes. These are like backups. If the holotype's missing, you can use the paratype plus, and then the others would become just the normal, the remainder would be the type series. Okay? So I would have holotype, paratype 1, paratype 2, and the rest are just specimens, but they're the part of the type series. So if this goes missing, you could use one of these. If these go missing, you use one of these. If they're all missing, you use a neotype. Okay? But if there's no holotype, then you just have syntypes. If you have holotype, you don't have syntypes. And then if all of these go missing, it's a neotype. But to do this, you would have a lectotype here, right? And then if they're all missing, it's a neotype. Does that make sense? So normally, in normal situations, if you're working with a new species that's never been described before and everything's okay, and you have more than 1 or 2 or 3 specimens, you're going to be doing this. Almost always. But if you have specimens that were described before or other things, then you start to go into these areas. There's a few other types, is it a hapentophyte type and a few others, but they're almost never used. All the other types are in the code, but the main types are these 5 types that I've introduced today. Holotype, paratype, lectotype, syntype, neotype. If you know those, you're fine. Sometimes you might see, especially in a plant paper, which is ICN or dinoflagellate paper, there are many other types. There's like 20 or 30 types. A lot of these are only for plants and a lot are for very specific situations. But I think for you, all of you in this class, it's probably going to be these. It's only going to be these 5. Make sense? Very exciting, huh? See, Heidi's trying hard to stay awake. She's like, okay. See if I just make sure I put the order wrong. I didn't think about the order too much. Holotype, syntype, paratype, lectotype, neotype. So there's other types too. So please see ICZN. This is it, yeah. Question.
How many paratypes do you use?
I usually do 2 or 3. I do 2 or 3, but you don't even have to make a paratype. A paratype is not a must, but I usually like to do 2 or 3. And I put the holotype, for example, if I find a species in Japan, I'll put the holotype in the National Museum in Tsukuba, and then maybe I put one paratype in Naturalis in Europe, and one paratype in the Smithsonian. But you could put them all in Tokyo too. But if you put, do you know the saying in English, put all your eggs in one basket? Yeah. So I like to put my species wider because it's becoming more international. But there's no rules about this either.
So in your case,
yeah,
you took the like huge colony and divided it.
No, because the types, I don't do subsamples because that's actually part of the holotype. So no, I don't do that. You don't make, you could for a colonial animal take different parts of the colony, but usually that causes confusion down the line. So I would take 3 different colonies, but I would know that they're in the same group, having molecular data or something like that. That's a good question though. I did have once, it gets confusing with colonial animals. And I see Kurt's got a question. I'll get to that in a second. There's a group of zoantharians, they're called Abyssoanthus, they're from the deep sea, and the polyps, they make a colony, but then the polyps separate. So each polyp is like an anemone, it's unique. And I had one reviewer, I had a rock with many polyps, and one reviewer said, you must choose one polyp as your type. And then other people say, no, it's okay. And So the rules are vague. And I would prefer to choose a colony, but sometimes the colony splits. What do you do? There's no rules. It's a tough question.
Yeah.
Yeah. Kurt, do you have a question?
I just want to like clarify for the syntypes and the paratypes.
Yeah.
So for the syntypes, you provide description as well together with the holotype, while for the paratypes, you just mention it. Like,
No,
so
no, no,
what's
no,
like
no,
the
no, no,
difference?
no. If you have a holotype, you do not have syntypes.
Oh,
Syntypes
okay.
only exist in older papers when a holotype was not designated.
So like in new papers, like right now, so you don't need to designate syntypes.
No, you should not. A syntype is basically it's a new definition to describe species when people didn't designate types at all, basically. No holotype.
Oh, okay. So right now, so
It's
if you have
solution
like a
to
lot
a
of
problem in the
samples,
past.
so if you have like a lot of samples, so that's a paratypes. The rest are paratype.
No, well, the rest can just be part of the type series. They don't have to be anything. A paratype is like a backup. So
But
you
like,
can-
take for example, like you make an original description of the holotype, and then you mention some
You-
other specimens that were
could
examined. So what do you call?
designate- So yeah, Kurt, you could designate 1 or 2 or 3 paratypes. And these are replacements for the holotype, just in case, kind of. They're like backups, but you don't have to designate a paratype. You could just have all the rest of your type series. They're just part of the type series, but they're not a type. They're just part of the type series. But if you have a holotype, you do not have syntypes.
Okay. Thank you.
That makes sense?
Yeah, kinda.
So para, if you think about the word para from Latin, it means alternate or like similar, right? So the idea behind a paratype is just in case if you lose the holotype, then paratype 1 should become the holotype. If you lose holotype in paratype 1, then paratype 2. It's like a backup. You, as the original describer, as the original authority, have designated these backups.
And these backups could be like placed in other museums.
Yes, they could be placed in other museums if you think that's wise. That's up to you. So
Thank you, sir.
no worries. I think it's good to put paratypes in other museums because of disasters and things. But are the paratypes the same species as the type? The holotype is another question, right? So it's it's tough. Okay, there's something in the chat here too. Hang on.
Nah, just me.
Yeah, okay. Kurt's mentioning, yeah, National Museum of the Philippines, collected by American scientists during when the United States occupied the Philippines, was destroyed in World War II. So a lot of museums have been destroyed. And so the further back in time you go, the less chance that you're going to find your specimens. Yeah, Guichet.
So in the case that you have, for example, your not yet your holotype, right? You didn't designate anything, but you're going to describe, right?
So before you describe a species, when you're working on the paper, yeah?
Yeah.
Yeah.
So you have a species that you say, oh, all this is one species, but they have maybe different colors, right?
I would be as safe as you can. I would be, if you're not sure, if there's even a little bit of doubt, choose the same color as much as you can. Choose the same everything as much as you can.
But if you are sure that they are the same, follow the same.
Yeah, but you still might make a mistake. Things will change down the line. I would be as conservative and as careful as you can in designating paratypes. Because you don't know what people in the future are going to find. And our idea of species will change too. The idea of a species is always changing, right? So people 100 or 150 years ago, they didn't make mistakes taxonomically because they were lazy or stupid. They made mistakes because they didn't have the data we have now. That's all. There's no other difference between them and us. So in 150 years from now, whether it's us or some ChatGPT version 200, someone's going to be doing taxonomy and they will look at our data as primitive and simple. So you just do the best you can and be prepared to make mistakes. Don't worry. Guichet.
But my question was, if you have to say like, oh, there are like color differences, do you put it in the holotype?
What do you mean color? If your type is your holotype's one specimen, there's no color difference.
Okay, so but you choose the blue one because you said this is the most abundant, for example.
Okay, but you should also put that information in there.
And but you also say that the color may vary.
Well, good. Some species have different colors,
Mm.
right? But that's case by case.
But for the paratype, you should choose the blue one.
I would.
Okay.
You can choose what you want, but if I'm on the paper with you, I'm gonna complain. Does that make sense?
Yeah.
Yeah, but again, I mean, look, what's important and what's not important are different in different species. And for some species, we don't know what's important. We don't know what the diagnostic character is. So you just do your best. That's all you can do. Okay. Other questions about types? Roger.
A quick clarification question.
Kurt, you got another question?
Is Roger finished?
Roger was telling us that you had a question. So yes,
Oh,
Roger.
okay. Arigato gozaimasu, Roger. I was just wondering with regards to the type.
Yeah.
Like it says here that a new type can only be designated when it's lost or destroyed. I was just wondering, can you designate a type if, like, for instance, like a type specimens has already been degraded, like has been described like maybe 200 years ago, like you want to designate.
Well, yes and no. You probably need to provide an explanation for why you're making a neotype. But if the neotype is there, but it's in formalin or was in formalin and you can't get DNA, that's usually not a good enough reason to make a neotype. You can try and explain it, but it's got to go through reviewers, and there's a chance that after that, still someone might complain anyways.
Yeah. Thank you, Sensei.
No worries. I know it's tough. It's tough. Old specimens are often in bad shape. Yes, Guichet.
Let's say that you're working on a species and you know the holotype is in London
And you can't go to London because it's expensive.
no, like you also find in Okinawa, right?
Yeah.
Wow, the holotype is in London, but I want to deposit in the museum in Okinawa.
Yeah.
What do you deposit? Like,
Wait. You're working on a species, but if the holotype's in London, it means it's already been described.
yeah.
Okay.
But you found it.
You found a new record.
Yeah.
So this is not a type. This is a simpler part of taxonomy. You find a new record of this species in Okinawa, you can put it wherever you want.
Oh.
Yeah. But if you're going to publish it, you need to put it into some kind of museum. But museums will usually take a type, no problem. They're very happy to take a type unless they have no money and they're like, this is scary. But for a new record or for just a lot of collection, many museums do not have space and will not take your specimens. So it's also local politics, how much space, how cooperative are the curators, et cetera. Maybe, like some places are very, very positive about taking in new specimens and others are not. And recently, there's more and more rules about international shipping and countries and permits and blah, 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 blah. It's messy. Case by case. Is that okay? Hi, Stefan, you had a question?
Yes, I have a question about the species where they have a very complex history of taxonomy.
Yes, messy history. Yeah.
And change family.
Yeah.
Does that, if you still disagree with it, does that mean you need to go through the list of holotype and paratype in every museum where they have
As much as you can, or you get people at the museum to take photographs and send them to you, or et cetera, or you have a big group of collaborators. That's why Zoanthus, 150 species, Paleothoa, 250 species, Acropora, 700 species. These genera are a nightmare to solve. Andrew Baird and his group are working on Acropora with UCEs, but to do the taxonomy, you have to go to all these museums, look at all the specimens. It's a lot of work. That's why the older, bigger genera with many species, those are the most painful. Very, very difficult. Yes. Good question. Any other questions online? Are we good? Okay, I've got a couple more comments very quickly. Actually, I didn't write it on there. There's another thing you can do to help future taxonomists. If you're working on a group and you've gone through all the literature and you have a lot of information and you're like, wow, this is really cool. You can do something called, right here. Called a nomenclator. Sounds like a robot or something, nomenclator. But this is a list of all the species or all the genera or all the families in a group you're working on. And it's got all the history, just like Stefan was saying. Putting together a nomenclator is usually it's a very taxonomic piece of work. But this provides information for people who are interested in taxonomy. I've done a nomenclator for Zoantheria for genus and higher. All the genera, all the families ever described, we have an 80-page nomenclator out for that. It was a ton of work. But for species, that's next. That's like hundreds, right? But you could make a nomenclator for the groups of animals you work on, and it's like a guidebook. It tells you all the names that have been used, whether they're valid or invalid, the history of them. Worms is kind of an online nomenclator, but it's not perfect yet because there's so much history for so many of these groups. But if you're really bored, for one, or if COVID-24 happens, for two, or if you really want to have a good piece of information for you for the future, make a nomenclator. Yeah, Gishe,
But this is,
yours is easy.
this is just a piece, like a list of species.
It could be a list of species, it could be a list of genera, it could be a list of families, but it's got the history of the names. And it's not only the valid, it's also all the invalid ones too. It's a list of everything that happened in that taxon.
But does it work well also as a by bibliography?
It's kind of like a bibliography, but you're putting comments in there too. You could say, Nanipura Kamuriya was described in 2015 by Miyazaki and Reimer. It was described as blah, 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 but then Dere said blah, 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 and then Dere didn't agree with blah, 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 and it's got all the information in there. Does that make sense? I've got one for genera and higher, and was so much work. It's a lot of work, but it's fun. So if you're a total taxonomic maniac, you can make a nomenclator. The flip side is if you are, for example, like Mari or like Kenta, or maybe like next year, like Dan or like Roger, you have a lot of data and you're trying to figure out what do I have? If someone else has published a nomenclator, it's really helpful for you because you can start to sort out all the different groups of your own animals. So something to think about. Crabs, crustaceans, mollusks, there's lots of nomenclators. Many other groups, nothing. Yes. Any other questions? Yeah, Gishe.
When you take
Mhm.
say, well, it can be anything, it can be species or genus, and you reclassify, right? Is it like, oh, we discovered that this doesn't belong here?
So you move a species from one genus to another.
Or genus to another
You
family.
move a genus to another family, yeah.
This needs to be published, right?
It has to be published, absolutely.
There has to be a paper.
Yeah.
Where it,
Yeah. Otherwise, it's just your idea. Yeah, everything has to be published. Otherwise, it's just an idea. Like, for example, like I've got, I think, an undescribed genus or two of Zoantherians, but I know they're new or undescribed, but I haven't written the paper. So right now they're nothing. They're just in my head. Has to be published. Okay. Questions? Anything else? I have a few last notes on taxonomy that I thought I should share with you guys. Okay. Okay. Let's go. Okay. So this is thanks to Kurt, but this is actually super important. Taxonomy really needs soft skills and soft power Some taxonomists do not have soft skills, and usually everyone hates them. I mean, in the end, you need to talk to people at museums. You need the people at museums to trust you. You need to talk to people going to the field. You need to talk to people running cruises or field surveys or expeditions. You need to talk to editors. You need to talk to researchers in other countries or grad students in other countries. All of these things are interactions with other people. And you need to be able to talk to other people, even people that maybe you don't agree with. And you need to be able to collaborate if you can. So I think not only do you need soft skills and soft power, you will do more as a taxonomist and as a scientist if you are collaborative, if you can work with other people. Not everyone can, and that's okay. But I know when you're working on a group of animals, you feel like they're your animals, right? These are mine. And then someone else appears and maybe they're doing the same thing. And your first instinct is to protect. And that's okay. You need to protect yourself to some degree. But 90 to 95% of people, this is my theory, 90 to 95%, 90 to 95% of taxonomists are good people. Some people are not. There are some, few, but there are some. Most people are good. And so I think, statistically speaking, most people are kind. Statistically speaking, you should give people a chance and work with people. And if you, especially with taxonomy, because there are so many undescribed species, so many taxonomic problems, none of us will ever run out of science. We will always have too much science to do. Yes, Chloe's like, oh no, think how busy you are now. You're only going to be busier. More questions, more ideas are going to keep appearing. And the only way that you can really solve a lot of these questions is working with other people, especially in taxonomy. And I think until now, some taxonomists have really not been collaborative. Some taxonomists have been really not kind. And I think this is one of the big problems with taxonomy is many taxonomists look at people who don't know all of these things we talked about, types, names, etc. And they just go, ha. The reason many people don't know taxonomy is because they never studied taxonomy. We should all be doing our part to show that this is a living, important science. Yes, and not just saying, ha, ha, ha. The last thing, though, is promote yourself at the same time. Yes, you need to work with people. Yes, you need to collaborate with people. But by promoting yourself, this can be, for example, emailing someone and saying, hello, I read your paper. I'm interested in this. Make conversations, start conversations, raise your hand. That's okay. As well, for example, I sent an email to some taxonomy related people in the lab. There's a top 10 new marine species of the world. Nominate your species. No one else will. So if there's an award or a scholarship, apply. If there's an award for some sort of like ICRS, I told you guys, ICRS has many awards. Nominate yourself. Ask someone to nominate you. No one is going to help you. You have to help yourself. You have to start the conversation. And if you have an award or a top 10 species or a newspaper article about you, that's good. It helps you. It helps your career. It helps you get funding, helps you go diving, helps you go to the field. So don't be on social media every day. Hey, new species, that, whatever you want. But you need to promote yourself because no one else will. Okay? I will always write you guys a reference letter unless I say I'm not writing you a reference letter. And that's very rare. I almost never do that. I think 180 people in the lab, I will write a reference letter for 177 of those people. 3 people I will not. So everyone here is fine, don't worry. But what I mean is you need to find opportunities and you need to find chances yourself and then ask people for help. Does that make sense? So, and soft skills, thanks, Kurt. And then this is something I've also thought, and promote yourself. The last thing here, this is a general rule for taxonomy. It's actually in the ICZN. It's not a rule, but it's a guideline. If during your research, you find out that someone else is working on the same undescribed species as you, ICZN says, wait a year. And if they haven't published, then you can go ahead. But as much as possible, contact the other people and discuss with them and maybe work together. ICZN, the most vague, ambiguous rule book in the world, says work together. If they say work together, you know, of course, right? Work together. So I've had cases where people were working on undescribed genus, 2 different groups. I was editor at one journal, Bert Hoeksema editor at another journal, same data, different research groups. So we got them to talk and they published together in the end. But we solved it would have been a huge problem. Maybe this paper one day earlier than this paper, good, invalid immediately. Total disaster. So you need to communicate with other nematodes or anemone people or polychaetes or isoamochi or sea cucumbers or nanipor or hard coral or, well, garbage is okay, but or zoanthariums. But you need to be communicating with other people in the field to know if what you're doing is unique, right? Okay. I think that's all from me for this class. I actually talked about everything I wanted to talk about today. Any last questions, comments, or anything? Iori-san.
Recently, I'm seeing the abbreviation of
Komnov.
Yeah.
Yeah,
What is it?
that's what Gisha was just asking me about. When you move a species to a new genus, or you move a genus, or no, when you move a species to a different genus, or when you move a genus to a different family, it's a new combination, novel combination. Combination novel, novo, new combination. So when, you know, say we move Palithoa umbrosa to Spinipus. The first time we write a paper, say we have genetic data, this is Spinipus. It's not, but if it was, it's Spinipus umbrosa Komnov. Okay? Okay, cool. Any other questions? Kenta! Hi. Yeah, Japanese, no problem. I'll translate it.
なんかちょっともしかしたら変な質問かもしれないんですけど、
大丈夫ですよ。
例えば新種かもしれないってなった時に、
ミキ採集。
ミキ採集だかもしれないってなった時に、まあ他にホロタイプとかいろんな他の近い種のホロタイプ見るわけじゃないですか。
はい。
その時にまあイソギンチャクだったらやっぱ内臓見たいって思うじゃないですか。その時にホロタイプをぶった切っていいのか、パラタイプをぶった切るのかっていう。
Okay. So Kenta says, for example, maybe you have something that you think is an undescribed species and you want to check some closely related types. Can you just go to the museum and start chopping them up, doing cross sections and stuff? Usually no. Sometimes yes. If you are a known expert on anemones, people might let you cut, but each museum has its own rules on what you can and cannot do. And usually there's paperwork that you have to sign and stuff too. So that's the other thing. If you have no papers and no publications and you say, I want to go to the museum and look at this type specimen, you probably need your sensei's help. But after you have a few papers, oh yeah, that's Kenta. He's a famous anemone guy. No problem. Let's let him do one cross section. Yes. Okay. Okay. Yes. But it's really group to group and museum to museum.
But regarding this, can you maybe let the museum or ask them if they want to do it for you?
You could, but usually museums are understaffed. And unless the person in the museum is an expert on your group, they're probably not going to know what to do any more than I would know for like a polychaete or something, right? Yeah.
So usually it's more common to go and work yourself with the sample rather than ask them.
External appearances, it's pretty common to ask for photos. But if you want to do some subsampling or actually do some morphological internal analysis, it's more common for you to go. But I wouldn't start touching any type of specimens until you know what you're doing. So that's also, you should develop your skills for working with your taxa. Cool. Anything else? Daijoubu desu ka? Okay, so Fiona is giving a talk at 3 o'clock in this room. Should be really cool. We've got an hour and 15-minute break. I'm gonna go do paperwork because I like paperwork. And I will see all of you guys at Fiona's talk or at the izakaya tonight, if not next week. Otsukaresama desu.
James,
Yo.
thank you for that.
Sure.
Kristen and Hiroki are asking if we can also do my talk over Zoom so they could.
Yeah, no worries. That'd be cool.
Can we just use the same link as now?
I'm probably gonna shut this down and go back to my office for a bit because we've still got an
Okay.
hour, 15 minutes. I'll make a new link. Hiroki and Kristen on the Mise chat, right?
Yeah, they'll get it.
Okay, I'll put it on the Mise chat.
Great. Thank you.
Thanks,
See you soon.
ciao, ciao, ciao. All right. Wow, everyone's fast today.
Isn't it?
ありがとうね。